All right, in chapter 5, I'm going to talk about the structure and function of macromolecules. And the main ideas here is that you've got compounds that consist of carbon, hydrogen, and oxygen, which are used to make carbohydrates, and they store vast amounts of energy in the case of like a lipid or a carbohydrate or something like that. Proteins have a wide range of functions, living organisms. I'll talk about that in a separate lecture from this one. And then the structure of DNA allows efficient storage of genetic information. So how that becomes the genetic information, how it codes for the proteins, that'll be yet another type of discussion. And we'll also be talking probably in a fourth one about lipids, okay? So one of the TOK questions that I want you to look at today here is there are conflicting views as to the harms and benefits of fats in diets. How do we decide between competing views? So take a second, write your answer to that down, and then we'll discuss it in class, okay? All right, so now as far as macromolecules are concerned, there's a wide variety of macromolecules that make up living organisms, and many of them are very large. Very large molecules consist of many hundreds or thousands of atoms to make these things up, and we call them macromolecules for that reason. There's 4 types of macromolecules that we're going to focus on in living organisms. We have proteins, lipids, carbohydrates, and nucleic acids, and each one of these will kind of make up a separate lecture for us. Okay, so 3 of the 4 classes of these macromolecules are made up or kind of comprised of polymers, and what that means is they're long molecules that consist of many subunits called monomers that get covalently linked together and make up these macromolecules. Okay, so carbohydrates, proteins, and nucleic acids are these polymers, okay, and the individual monomers for like a carbohydrate would be a monosaccharide, for a protein it would be an amino acid, and for a nucleic acid it would be a nucleotide. Okay, so we'll kind of get to that as we go through these. All right, so we talked in class about the synthesis and breakdown of macromolecules and how they're governed by a series of buildup and breakdown reactions. Of course, the condensation or dehydration reactions are the ones that are going to build them up. And then the hydrolysis reaction, if you recall hydrolysis, using water to break the bonds, that's what we use as the breakdown types of reactions. Okay, so these condensation reactions, these are where monomers get added to this growing polymer. And during that process, we went over this in class and we showed how a hydroxyl group on one atom reacts with either a hydroxyl or a hydrogen atom on another molecule, and H2O is given off. And these types of reactions, whether it's a dehydration type of a reaction or a hydrolysis type of reaction, they require some energy, and the action is carried out by enzymes to kind of help lower that activation barrier, which we'll talk about in chapter 8, a later chapter. Okay, so here if you look at this, you can see the short polymer there at the top. You can see the hydrogen on the one end, and then you can see the unlinked monomer there, the hydroxyl. And we went over this exact same thing in class, and when we link those together, the help of an enzyme, we're giving off the water molecule, and that's why it's called a dehydration type of a reaction. Okay, so hydrolysis now, again, hydro meaning water, lysis meaning to break apart. We're using water and the help of an enzyme to break these atoms or break these monomers apart. Okay, so this is the type of thing that would occur during digestion. So you eat some food, consisting of all of these polymers. The enzymes that are getting released, these are the things that are going to split up and break these monomers into the individual, or break these polymers into the individual subunits or the monomers, so that our body can use them for growth, repair, energy, that type of a thing. Okay, so we can take these 4 types of macromolecules and we can break them down further, in terms of what we're going to do here shortly. So here's your large polymer there at the top, and you can look at the help of an enzyme and a water molecule, breaks that apart and yields these individual monomers. Okay, so carbohydrates now, getting into this a little bit more. These are large macromolecules consisting of carbon, hydrogen, and oxygen. In the 3 classifications which we went over in class, we talked about monosaccharides or single sugars. Okay, sometimes they're referred to as simple sugars. We have disaccharides, which are 2 individual monomers linked together, or a double sugar. And then we have polysaccharides, which are sugars or large macromolecules, carbohydrate macromolecules consisting of 3 or more carbohydrate monomers or sugar monomers. Okay, so you'd have like starch or cellulose or something to that effect. Okay, so with a monosaccharide, the general formula for this would be CNH2N ON. So if N was 6 there, you'd have C6 C6H12O6, because the 2N means multiply the number, whatever N is, by 2. So glucose, C6H12O6, that's a common sugar that you're probably familiar with. And this is used very readily by our bodies as an energy source. So you can look at, there's various forms of the way in which glucose is put together. So if you look there at the figure on the right, you can hopefully guess what type of a sugar molecule that is, which type of glucose that is. And if you want to know, ask me in class and I'll be happy to tell you. Okay? So the monosaccharides here now, we don't, just use these as an energy source in terms of immediately, but they serve as the kind of the carbon backbone or the carbon skeleton for larger macromolecules. So within our bodies, for instance, if we're taking in these sugars, we can use them if we need to make them amino acids. We can use them for that. We can use them to make fatty acids. We can, use them for a variety of different things. So they're not just an energy source. Our body uses them for all sorts of things. The disaccharides, a very common one that we talked about in class was sucrose. The other very common ones are lactose and maltose. Of course, sucrose, that's like the white crystal sugar that you put on cereal if you eat it, or you cook and bake with it, things like that. Lactose is milk sugar, and then maltose is just a disaccharide of glucose molecules, and as many of you pointed out in class, it's used to brew beer and things like that. Okay, so what you have when you join these, whether it's a disaccharide or a polysaccharide, you get these glycosidic linkages. So for instance, with the activity that we did in class with the two different molecules, when you take the number 1 carbon and the number 4 carbon, and you guys linked those together, what you were forming was a glycosidic linkage. Okay, and that's the covalent bond between the two monosaccharides using a dehydration reaction, because of course you gave off the water molecules, which you all built as well. Okay, so the fruit of a plant is a great source of sucrose. We talked about sugarcane being another one, and basically what sucrose is, table sugar, is glucose and fructose linked together. We talked about the sugarcane processing, and when you go through all that, there's a leftover, and that's what forms molasses. And I showed you what the molasses looked like, that dark brown sugar. That's kind of the leftover after the sugar processing. If you bleach it out and process it, that's the white crystalline sugar. If you process it minimally, that's kind of the sugar in the raw. It kind of has the brownish color to it. And processing it even less would give you the brown sugar. So it's got a little bit more of the molasses stuff that you're taking out. But the sucrose, this forms during the plants when they're performing photosynthesis, and then when it gets stored, it's often stored as a sucrose molecule, and then we extract that for our purposes of cooking and baking and things. Okay, lactose is another common sugar. That's a milk sugar. That's glucose and galactose. So if you look at the disaccharide on the left at the bottom there, that would be an example of what lactose would look like. Maltose, this is kind of an amylose type of a sugar, a wheat sugar type of a thing. So if you look at the action of amylase, which is a common enzyme either in your salivary glands or from your pancreas, when that's breaking down starch, maltose might be one of the breakdown products. And if you look at the bottom right figure, for instance, that would be an example of maltose. Okay. Now, polysaccharides, we said that when you form these glycosidic linkages between these individual monomers thousands of times, this is where you're getting this polysaccharide. And there's two main types that we talked about in class. We talked about structural and we talked about storage. Okay, so the two main forms of glucose, we have alpha glucose and beta glucose, and we did a whole activity with that. And it's all determined by the placement of that hydroxyl group. Okay, so if it's below the plane, you look on the left there, that would be the figure would be alpha glucose. If you look on the right, where hydroxyl on the number 1 carbon is above the plane, that's where you've got beta glucose. And if you look at the little figure here on the right that's moving, that's going from the linear structure, which you all looked at, and then as it's getting bent around, that's when you're getting either alpha or beta glucose. And it just depends on what's going on really at that number 1 carbon when it's spinning, which side is it ending up on. Okay, and you'll either get alpha or beta glucose. All right, so now whether it's structural or storage, we talked about the structural types of polysaccharides when you've got these beta glucoses getting linked together to form the cellulose. You guys made one of those molecules in class. We refer to that as dietary fiber. This is forming the cell walls of the plant to give them some structural rigidity because they don't have skeletons, for instance, like we do. And the plant cell walls, that's what we refer to as insoluble fiber or dietary fiber. And you find it in a lot of fruits and vegetables, we said. And we said that there's some animals, such as ruminants, like cows, that can digest the cellulose, but humans can't. We lack the enzymes. We lack any sort of organism, perhaps living inside of us, that could do that. But cows have, a different type of a digestive system, and they're able to eat the plants and break down the cellulose and extract energy from it. Okay. Another important structural polysaccharide is chitin, and we talked about this being the hard exoskeleton of arthropods. It makes up the cell wall of fungi. So for instance, mushrooms and other fungi have different kind of texture to them, and that's because of the chitin, that structural polysaccharide that's found inside of them. Now, some storage types of polysaccharides: we have starch and we have cellulose. And you have different types of glucose, of course, that makes them vastly different. The alpha that's found in a starch versus the beta that's found in cellulose make it, so again, we can digest starch as an energy source, but not cellulose. Okay? So there's two main components now to starch. You have amylose and you have amylopectin. The amylose makes up about 20 to 30% of the structure and is a little bit more resistant to digestion. For instance, if you look at the bottom left, that's amylose. And what you get is these things really get kind of packed in tight, and it makes it hard for the enzymes to get at them, and that's why they're more resistant to the digestion than the amylopectin, which is on the bottom right figure here. And that makes up about 70 to 80% of the starch, and it's a lot easier for our bodies to digest because you can see it's kind of branched, so it doesn't pack together as much, and it's more available to enzymatic type of a digestion, and just, it has more surface area. It's a little bit easier to break down. Okay, and when you're looking at the two types of storage polysaccharides now, starch being one that we just talked about, and it looks at the amylose here at the left figure. When you're looking at that one, you can see the amylose, those chains that are not branched, versus the amylopectin that is. But the other form, the other storage form, which is found in animals, is glycogen, and this is very branched. It's made up of these glucose molecules that our bodies, of course, can use. We said that they're stored in liver tissue, they're stored in muscle tissue, and we talked about the whole idea of the fight or flight, being able to very quickly use this energy in our muscles, and also just when we get hungry, the whole idea that our pancreas releases glycogen due to the low sugar levels. That acts on the liver to release the glucose from the glycogen. So the glucagon breaks down the glycogen stores in the liver to bump up the glucose, the blood glucose level, to kind of keep us, from getting drowsy, to keep our brain working, etc. And we said that we'll come back to that more in topic 6 when we talk about the human body stuff. Okay, so the next talk that we'll go through next time will be on proteins, and that's the one that we started in class just recently. Okay, so we'll see you soon.